Hi, my name is Kylie Nee, and welcome to my presentation on the digestive system. Today I'm going to be talking about the regulation of the gastric phase. So for just a little overview about what we'll be talking about, so there's three major stages in the gastric phase. So the first is storage, which is when the stomach stores food and regulates passage into the small intestine, where most digestion and absorption actually takes place. And then there's protection and digestion, when the stomach chemically and mechanically digests food into a soupy mixture of uniformly small particles called chyme. And then there's defense, which destroys bacteria and other pathogens that are swallowed or trapped in mucus. So for storage, it's probably the least obvious aspect of digestion for people to think about. So both parts of the stomach are actually in use. So in the upper portion of the stomach, there's something called the receptive relaxation that's taking place, which is when the upper half of the stomach is relatively quiet and it's holding food until it's ready to be digested. And this allows the stomach to regulate the rate at which food enters the small intestine. So the stomach will relax and then expand to hold that increased volume of food. And then this regulation will allow digestion and absorption to be possible, and it allows optimal chyme to pass into the large intestine, because too much chyme production would actually lead to something called the dumping syndrome, and this is when too much chyme gets passed into the large intestine, which is not apt for large-scale nutrient absorption, and then the chyme gets turned into feces. So too much chyme obviously equals too much feces, and this results in diarrhea. So that's the upper portion of the stomach, and then in the lower portion of the stomach, it's actually very busy with digestion. There are peristaltic waves pushing food through the stomach. So these series of waves push food through the pylorus, mixing the food with acid and digestive enzymes, and large particles of food are digested into chyme, and then each wave squirts some chyme through the pylorus and into the duodenum. This enhanced gastric motility is under neural control and is stimulated by the distension of the stomach. So that's the storage phase. You can kind of see over on the right this figure. It kind of shows exactly what happens in the storage phase, and then the reflexes that take place after. So then there's protection and digestion. There's lots of different secretions that happen in protection and digestion. So in gastrin secretion, it has to do with the G cells that are found deep in the gastric glands, which secrete the hormone gastrin into the blood. And in short reflexes, gastrin release is stimulated by the presence of amino acids and peptides in stomach and distension. So a fun fact, coffee, whether it's decaf or not, actually stimulates gastrin release. Excuse me. And this is actually triggered by neural reflexes. So short reflexes are triggered by gastrin-releasing peptide, or GRP. And in cephalic reflexes, the parasympathetic neurons from the vagus nerve stimulate G cells to release gastrin. And then there's acid secretion, which have to do with the parietal cells that are deep in the gastric glands, which secrete gastric acid, or HCl, into the lumen of the stomach. And then this gastric acid has multiple functions. So it causes release and activation of pepsin, which is an enzyme that digests proteins. I can't say enzyme. The acid also triggers somatostatin release from D cells. HCl also denatures proteins by breaking disulfide and hydrogen bonds that hold the protein in its tertiary structure. And then unfolding this protein chain makes the peptide bonds between amino acids more accessible to digestion by pepsin. Gastric acid also helps to kill bacteria and other ingested microorganisms, and it also inactivates salivary amylase, which stops carbohydrate digestion that began in the mouth. There's also enzyme secretion that happens in this stage of the gastric phase. So in enzyme secretion, the stomach actually produces two enzymes, pepsin and gastric lipase. So pepsin carries out the initial digestion of proteins, excuse me, and it's particularly effective on collagen and therefore plays an important role in digesting meat. And it's secreted as an inactive enzyme, pepsinogen, by chief cells in gastric glands. And acid stimulates pepsinogen release from chief cells through a short reflex, which is mediated in the enteric nervous system. And then there's also paracrine secretions. So paracrine secretions from the gastric mucosa include histamine, somatostatin, and the intrinsic factor. So histamine is a paracrine signal that's secreted by this crazy cell called the enterochromaffin-like cell. And it's in response to gastrin or acetylcholine stimulation, and histamine actually diffuses to its target, the parietal cells, and stimulates acid secretion by combining with H2 receptors on the parietal cells. The intrinsic factor is a protein that's secreted by the same gastric parietal cells that secrete acid. In the lumen of the stomach, intrinsic factor complexes with vitamin B12, which is a step that's needed for the vitamin's absorption to the intestine. And then somatostatin, or SS, is also known as hypothalamic growth hormone inhibiting hormone, and it's secreted by D cells in the stomach, and it's the primary negative feedback signal for gastric phase secretion, and it shuts down acid secretion directly and indirectly by decreasing gastrin and histamine secretion. So over in the left picture, you can see acid secretion in the stomach and how it works. So actually, in acid secretion, it's at the parietal cell pathway for acid secretion that's depicted in this figure. And this begins when H+ from water inside the parietal cell is pumped into the stomach lumen by H+, K+ ATPase in exchange for K+ entering the cell. And then chloride follows the electrical gradient created by H+ by moving through open chloride channels, and then the net result is secretion in HCl by the cell. And then over on the right, you can see what's going on with arrival of food in the stomach. So in the first stage, food or cephalic reflexes initiate gastric secretion of gastrin, histamine, and acid. And then in the second phase, gastrin stimulates acid secretion by direct action on parietal cells or indirectly through histamine. And then acid stimulates short reflex secretion of pepsinogen. And then finally, somatostatin release by H+ is the negative feedback signal that modulates acid and pepsin release. Excuse me, I'm sorry. So then finally in gastric phase is defense. So mucus cells on the luminal surface and in the neck of the gastric gland secrete mucus and bicarbonate to make a barrier. And this is called the mucus bicarbonate barrier, and you can see it over on the right. And this is a physical and chemical barrier. Physical because of the mucus, chemical because of the bicarbonate. So when mucus secretion is increased when the stomach is irritated. So if you take too much aspirin or if you ingest too much alcohol, mucus secretion can be increased because the stomach is being irritated. So if the mucus bicarbonate barrier fails, this could result in hyperacidity. And there are some different types of things that can happen with hyperacidity. So one thing is called the Zollinger-Ellison syndrome, which patients secrete excessive levels of gastrin, usually from gastrin-secreting tumors in the pancreas, and as a result, hyperacidity in the stomach overwhelms the normal protective mechanisms and causes a peptic ulcer, and then in peptic ulcers, acid and pepsin destroy the mucosa, creating holes that extend into the submucosa and nucleolus of the stomach and duodenum. There's also acid reflux, which is a little bit more common, and that's which happens in the esophagus, which can erode the mucosa layer there as well. So that's defense. And then this is just a slide showing the secretory cells of the gastric mucosa. So you can see the different cells and what takes place in each cell. So in the mucus surface cells, mucus is obviously secreted, and this is stimulated by the irritation of mucosa, and its function is a physical barrier between the lumen and epithelial, like we talked about. Parietal cells, like we talked about, the substances that are secreted are HCl and intrinsic factor, and they're stimulated by acetylcholine, gastrin, histamine, and they are to activate pepsin and kill bacteria, things like that. G cells down at the bottom are secreted by gastrin, and they're stimulated by acetylcholine, peptides, and amino acids, and this is to stimulate gastric acid secretion. So that's just like a little overview slide of all the different cells that are affected. So then after leaving the stomach, the process of digestion then moves from the gastric phase into the intestinal phase, which I am not going to speak about, but thank you for listening to my presentation. Thank you. Bye.